Well, thank you. So hopefully everyone can still hear me okay. Thank you very much to the organizers for inviting me here. It's kind of like the Holy Grail for a mathematician to give a talk at the Fields Institute. So I really appreciate that opportunity and just to be here with all the new friends. And thank you everyone for online for joining as well. I'd like to update, and I want to point out there's a lot of joint work with my friend and cohort and colleague in crime, Alana Faredig. So I'll present some of this work here today. I'm going to put my thank yous at the beginning because we often don't get to the thank yous, but thank you to funding over the years from different agencies and some from nonprofits like the Jane Koskinas Teddy Violence for Health Foundation for Health and Policy. That's a mouthful. And also more recently, actually, as of yesterday, from the NCI. So we're really excited about that one. And we're really grateful to have a network of collaborators who've really been pitching in on this work over the years. Particularly Lana Fairdig's lab at Johns Hopkins and Genevieve Stein O'Brien and some of the others, Oregon Health and Science University, Indiana University, of course, my lab has been really phenomenal to work with. And, taking maybe just a little bit step back, I love the way that Thomas framed everything yesterday, kind of going through the hallmarks, and really bringing it back down to what the cells are doing. And to me, that's been a big motivator. I did my PhD work in moving interface problems, sharp moving boundary problems, where John Moengrab started with moving soap film interfaces that crossed out the word soap film and wrote the word tumor in. And that was our start using Darcy flows. And that was a really fun PhD. And then I got to MD Anderson Cancer Center and looked under a double microscope with the pathologist said, oh, right. And so basically stopped teaching methods at that point and went to discrete agents, particularly was really intrigued by all the immune cells that were swarming outside the breast ducts and saying, what's going on there? There's got to be something interesting. And then that was too scary for us to touch for about 10, 15 years, but we're finally kind of getting back to that. So really looking at individual cell behaviors to me has been a motivator, kind of an intermediate scale that you can connect upward and connect downward reasonably well. That in any tissue ecosystem, in particular cancer, it's neat to me that you start with a relatively small palette of behaviors. Cells can divide, they can die, they can differentiate and change type, they can consume things, they can secrete things, they can wander around, and motility, and of course there's variability, we talked a little bit about yesterday. And then you have pairwise interactions. The cells can stick to each other, they can push each other around, they can eat each other, which is rather fun. They can attack each other and cause damage. They can fuse together. That was a new one to me in the COVID era. Of course, most of us do that too. And they have other contact communication, like say delta notch signaling, so they can pass information over contacts. Then you put them into an environment that is full of physical constraints, that chemical factors and building supplies like oxygen and glucose, waste products like lactates, all have to move by diffusion, typically. I mean, of course, there's other transport processes, but they have to move by transport. And that means you have diffusion limits, like we saw in the earlier talks today with the diffusion length scale. And of course, there are mechanical barriers. And so this limited palette of individual behaviors, not humongous palette of cell interactions when you think about it at the cell level. Molecular soup level is a whole other story, right? That's how you do these things. And in physical limits, somehow, when you put it across many cells interacting, you get cancer ecosystems with very complex emergent behaviors. So I'm really interested how you start with these basic building blocks and implement those building blocks as kind of like starter points, and then write the rules of when you trigger these behaviors and interactions and how that leads to cancer or organized tissues or immune systems or other things. So to me, multicellular systems biology is really understanding how you start from these building blocks to the behavior of multicellular systems. And multicellular system engineering says, if we think we know what we're doing, how do we manipulate these individual behaviors and interactions or placement of, say, diffusing signals to push the behavior in the direction we want to go. So this is a nasty, complicated system. So like everybody, we use models to try to detangle them. Another thing that was eye-opening to me is being insulated in the math world as a PhD student, I thought all models were mathematical. What else could there possibly be? And then you start talking to biologists, oh, there are mice, there are petri dishes, there are organoids. Like, oh, I didn't realize a model could mean so many things, right? Now here we really do need mathematical and computational, but it's worth remembering that a model is a virtual laboratory, a simplification of reality where you hope to gain some insights, right? And so that's really what we want to develop. So to get towards that computational mathematical model, I want to take just a brief step backward towards a conceptual framing that I think is really helpful for cell interactions. And so I like to think of it as a signal response communication, even for mechanical signals. That you have two cells interacting, one of those cells is a sender of a signal, and the other cell is a recipient. And so a signal can be something that can elicit a behavioral response, so stimulus. It might be a chemical signal, it might be physical contact, it might be mechanical pressure, for example, or contact with a specific cell type. But you can interpret many interactions in this kind of a framing. A macrophage, a sender, secretes IL-6, that's the signal, diffused into the environment. It drives chemotaxis, that's response, by a CD8 T cell, that's recipient. So you have a sender, a message, a recipient, and a response, signal response. And so a lot of modeling can be decomposed into these kind of things. So you can say who's sending signals, who's secreting a chemical factor, and who's receiving them, and how do they respond to them. And so kind of take these into separate pieces. It makes the modeling a little bit more tractable when we start trying to combine complicated systems. And so agent-based models can be a very good framing for this, even just as conceptual framing. If you never simulate your agent-based model, but just take the time to write down the rules, that actually gets you a long ways towards understanding your problem. It actually becomes a knowledge model to help you understand and frame what you're looking at, and gives you a language and a context to talk to biologists and people in other fields. So agent-based modeling, which you've seen here and there over the past day or so, is really a discrete form of modeling where you say, I'm going to model a cell as an individual entity and has a combination of things. It has member data, if you're an object-oriented programmer. It has variables that are attached to that object. And they move around with that object, and they have their own individual copy of those things. Things like, what type am I? What's my position? What's my velocity? What's my humongous vector of my phenotype representation, like phenotype parameters? Any custom variables you might want to assign. And then on top of that, you have processes assigned to those agents, things that can basically act upon those parameters or use those parameters. So division, for example, would take my cell parameters and copy into a duplicate cell and pass some of those parameters to the new cell you've just created, for example. Death might interpret your death rate and decide whether to kill or remove the agent from your model. Migration is a Brownian motion, for example, with a bias term. So you can do a lot of your things saying, from my given parameters, I can control these individual processes that I've implemented. Remember that handful of palette of stuff that the cells can do. And you kind of template that in a very general sense, and then the individual cells can control the way they do these things and when they trigger them. So there are lots of forms of agent-based models. There are a lot of great people in this room who've built them. Costi has built these beautiful immersed boundary models that I'm just absolutely in love with, that go really just gorgeous cell morphology. So really, it kind of goes to resolution and alignment. That you can either divide your space up to grids into a big grid and say that each grid spot has one cell, and you kind of slide them from spot to spot. And if you do one cell per grid, you're in a classic cell automaton. I can move left, right, up, down, in and out, I guess if you want to do the Z direction. So I'm trying not to do Z, but, or I can divide and place a daughter cell in a neighboring grid spot. So that's kind of the classic cell automaton, hugely popularized at Moffitt, for example, and they did really, really well with HAL. If you want to kind of zoom out, you can divide that up into bigger lattice size and say, I don't care about where the individual cells are anymore. I just say that in this box, there are a bunch of cells and might have a flux to the next grid, and that's a lattice gap method. So less than one cell per grid. Or I can zoom in and say, now I care about morphology. So I'm going to divide it into a finer grid and say the individual pixels are bits and pieces of cells. So I swap around by an energy minimization, and that's a cellular pulse method. And so like James Blazer's CopyCell3D is a great example, or Morpheus is another wonderful example of that. Roland Merckx has great software there. So those are lattice-based, and those are great, but sometimes it can be very hard to come up with the rules of how to move cells from one voxel to another, particularly with mechanics. It can be very hard in those. And if you're in cell automaton context, it kind of assumes that all cells are the same size, which is a bit problematic. So you can do off-lattice models, where you say, no, I'm not going to restrict them to a lattice site, but just say I have a position, I might have a size, and I do a balance of forces act upon the cells, and that leads to a resultant motion of the cells. And so you can do things like potential functions to degree adhesion and repulsion. We steal that from molecular dynamics. Or you can zoom in, just like in the grid-based, you can zoom and say, rather than one agent per cell, I can do many agents per cell, and use potentials for them, and you get cell morphology again, but without the lattice. And that's called a subcellular element model. Or I can say, I'm going to do something fancier and model like the boundary points with either an inverse boundary method like Hosse's wonderful work, and I hope she'll show some of that, or other methods like vertex models that can track cell morphology. So ways to kind of take it away from the grid, and they have pros and cons. This off-lattice stuff is more computationally expensive, but more detailed. This stuff is particularly fast, but sometimes a bit ad hoc and hard to calibrate. So it plots this beautiful morphology, but then you have energy terms that are hard to connect to physical properties. And you have they often end up being correlated in that Hamiltonian. So you change motility, and all of a sudden it's also changing proliferation because it's connected in minimalization terms. So you have to be careful with those. On the other hand, these are a bit more physical, but they're more computationally expensive. So we'll be talking kind of right here in the middle of this, which is kind of a trade-off between detail and tractability. Oh, and we wrote a review on these several years ago. That's quite all at this. I can share a PDF of these to get the hyperlinks for references if that's helpful. But we tried to kind of go through a whole bunch of examples of the different approaches to age-based modeling in the cancer context and some of the key results. Angiogenesis, some beautiful work coming out of Moffitt and others, for example, for a long, long time. So we have these different representations of cells, on lattice, off lattice, resolution. But then we have to place those cells into a virtual environment because we care about the environment, not just the cells. We care about diffusing factors, we care about boundaries and membranes and things like that. And so basically, you couple your discrete agents with typically PDE models of chemical transport in your environment. So you need a combination of diffusion solvers and agent solvers, and then you have to figure out how to couple this in time. And within that context, then, cells become signal processors. My cell and my location has access to a broad variety of signals, things outside the environment like chemical concentrations, gradients, contact with other cell types, and things inside like, am I alive or am I dead? Am I cycling or am I not? What are my mutation rates? What are my transformation rates? What are my other properties that I've stored? And based upon a combination of external and internal signals, I can modulate the process parameters of each of these processes that drive the biology of that cell. So there's actually a great, I think, analogy to continuum models. And continuum models, you start with kind of a general form, like conservation of mass, conservation of energy, conservation of momentum. And that's general. And we say, but I want to talk about Darcy flow. So you have a constitutive law that says that the velocity is proportional to the pressure gradient. And that specific form, plus say an incompressibility assumption, goes from the generic functional form to the specifics of your particular problem. In agent-based modeling is the same. We have general process models for cycle entry, Brownian migration, adhesion, secretion, source terms. That's generic. And then your constitutive laws are biological rules or hypotheses that say, for this kind of cell, I trigger division in this way after these signals. I trigger motility up these gradients. So going from the generic process models to the specific becomes a constitutive laws that are codified as rules when you do agent-based modeling. So there's that analogy to continuum, I think, here. The biological hypotheses are basically your constitutive laws. And ABMs have a little bit of stochasticity to them, which can be very fun. Just some classical examples. Proliferation. You might have a proliferation rate or cycle entry rate R, and you might vary that R. Any parameter, beautiful thing is you can vary it in space and time based upon the signals. So for my instant in time, for my cell, I have a particular transition rate or cycle entry rate. And so I say the probability going from entering the cell cycle between now and t plus delta t. So this chunk of time delta t is basically just the rate parameter times delta t. And the probability of death is very similar, right? So you basically say, at my point in time, what are my parameter values now? Use those to calculate the probability to decide if I'm going to do something. So that's a discrete calculation. Migration is also very fun. I might say that I have a bias direction, say a chemical gradient this way. And then I have a bias parameter between 0 and 1. If my bias parameter is 0, I ignore that, and I just pick a random vector, and then I normalize it, and that's my direction of travel. And then I go for persistent time, stop, and pick another direction. So if you have bias 0, you recover classical Gaussian motion, Brownian migration. If your bias is 1, then you ignore the random term, and you just say, my direction is up that gradient, and then you normalize it, multiply it by a speed, and then basically get classical chemotaxis deterministic. And then the more interesting ones in between, say maybe I'm 50% biased. So I pick, that's the way I want to go. Pick a random vector, I merge them together, normalize it, and that's the result direction to go. And kind of pick a weavy path as a biased random migration. So you can recover classical chemotaxis and everything in between with this kind of a functional form. Transformations from type A to type B, say from monocyte to macrophage, for example, or other differentiation pathways or mutation pathways, all stochastic, transitions. And so, Mark.
Just curious about that.
Yeah, it's really warm in here. I'm going to get rid of this.
How do we choose that bias parameter? Do you have some experimental data?
How do you choose the bias parameter is the question from Mark, and the great question. The answer is wisely. So that's not helpful. So what we've tended to do is use approximate Bayesian computing to calculate those. And so we have a persistence time. What we tend to do is say that there's no point in setting persistence time too fast compared to my sampling resolution. So if I'm imaging every 5 minutes, I'm going to pick a 5-minute persistence time to kind of match the resolution of my trajectory so I can measure. So fix usually my persistence time. Linear speed is easy because you just track that. As long as you want the parameter, the bias parameter. And so you can use approximate Bayesian computing to compute a bunch of cell traps and compare that. So that's an example. So kind of connecting this to the hallmarks of cancer, every one of these things can basically be addressed by trying to modulate one or more parameters in one or more cell types. Angiogenesis is over here somewhere. Beautiful work going about 20 plus years on that, right? Taking discrete endothelial cell models, saying I move and move and move, sometimes divide and extend a trail, then I might connect and form something, doing anastomosis, responding to increasing the secretion of VEGF by other cells. So I have one cell that says, in low oxygen, increase secretion of VEGF. And then you have another cell that says, when VEGF goes up, break away my adhesions, start migrating. And then if I'm, depending upon what I'm doing, maybe I migrate, maybe I divide and leave a trail of endothelial cells. So for instance, an example, right? Immune walls, activating invasion metastasis, you might want to change your cell adhesion, change your migration characteristics, probably do some ECM remodeling, both by you and other cell types, like say fibroblasts. So all these hallmarks can be addressed if you start thinking about the rules of them. And notice that a lot of them basically have the same approach, just like we had in Thomas's talk yesterday. It's hard to tell the difference on how to model phenotypic plasticity, epigenetic changes, and just variability, because they all end up just saying you probably should vary some of the parameters randomly in time. And I think there's some nuance to be had and some consensus to be had on how to do that and what would be a good representation of that. But you can address these. So it's just a fun starting point to say, because I have all these behaviors can modulate, I can say which behaviors are associated with different hallmarks of cancer and come up with the rules of them. And that would be kind of our starting point. So for our virtual laboratory, we said, well, diffusion is pretty important. We started off modeling a lot of hypoxia because that was our original interest, but we knew we'd have to do hypoxia, maybe some metabolites, a few growth factors, maybe a drug or two, and you're talking 5 or 10 diffusion equations, easy to do an interesting problem. And back when we started, everyone was modeling one diffusion equation at a time, typically handwritten in some code. And so we wrote a finite volume method that vectorizes a class of diffusion decay equations. It solves them vectorially, basically a big tensor thing before we were cool and call them tensors. But we do a local one-dimensional method, solve a vector of X diffusion, vector of Y diffusion, vector of Z diffusion. So it's like an operator splitting. The conversion is nice if it's easy to do. It's the Dave Thomas, sorry, not Dave Thomas, where's the P? Just the Thomas algorithm. The forward sweeping back substitution, because it's tridiagonal, it's a vector tridiagonal systems. And the cool thing is that if your diffusion and decay constants are constant, then the operator part of your linear system is unchanging. So you can pre-compute and cache the forward sweep, and all you have to do is calculate the back substitution. So that cuts your compute time in half. So lots of cute things like vectorizing and cleverness allowed us 10, 15 years ago to solve like 5 or 10 diffusion equations in 3D on the desktop. So it's a start. But that had cells basically as point sources and sinks being there and being stupid things. There's no biology in that yet. So Physicell took that as a base and said, now let's add some biology to those cell sources and sinks. We added the cell cycle state, depth parameters, built-in migration, secretion sources, And over the years, we've been adding more and more to it based upon the demands of the modeling community and the problems that we encountered, particularly in COVID, when we started adding immune behaviors. And so this is open source a while ago, published about 5 years ago. It would have been maybe 6 years ago, but peer review was interesting. And this works on Linux, on Mac, on Windows. You can run it on your desktop, and when you're ready to send it over to a compute cluster and run 100,000 copies of it. So it's nice that it's not connected to a GUI that you have to use, but lately we're developing design tools. So here's just an example where you can see the heterogeneity they can do in cancer and the variability. I start off with just a really simple system. Each cell has got this initial salt and pepper distribution of just, let's say, an aggressiveness parameter, call it an oncogene if you want to. Blue is less aggressive, yellow is more aggressive. All of them require energy, so we use oxygen as our energy proxy. Boundary condition, Dirichlet diffuses in, every cell consumes it, so you get a gradient of oxygen as you go in. So the more oxygen you have, the more you proliferate. And if you're yellow, you do it better than the blue cells. And that's it on there. And it's heritable. So daughter cells get the same property as parent cells. So you'll see a selection process. When oxygen gets too low, you will see cells become brown and necrotic. So they will shrink away but maintain adhesion. So you kind of get an instability mechanically there.
Sorry, when cells divide, what happens? I mean, what's going on with?
So when we do division, basically, a great question. So when we do division, we take the original parent cell diameter and we conserve the volume. And so the two daughter cells fit in the parent cell volume with random orientation right now. And then the potential functions will push them back out and reshuffle the neighbors. And so we also have a continuous growth process for all the cells. So it's not quite such an abrupt thing. So they'll divide in half and then gradually push each other away and gradually grow back towards the parent size over time. And so that turned out to be numerically smoother and more realistic. So it was a win-win than just abruptly doubling it in volume at division, like older models. That used to require some weird adaptive time stepping to make it work well stably. And the fun thing is we tried to be clever and say that you would adapt your delta T based upon the velocity of the cells. So you'd hum along with a high delta T, and then a cell divides, and then two overlaps. They have high velocities from the potential function, so you drop your delta T. Then they adjust, and you go back up to V. But what happens in cancer? You're dividing all the time, so you end up stuck at your minimum delta T all the time. So it wasn't worth, clever was sadly not clever. So I just want to give an example before we kind of get into the bits of the talk. So this is called hypoxic adaptation. I should have probably shown actually what happens in this thing. I apologize for that. Way to take the sizzle out of it. So the dividing, you can see the yellow cells are begetting. Yellow begets yellow, blue begets blue. Seems kind of biblical with the begetting. You can see this selection for the yellow cells because they're out competing the blue. When you have the combination of opportunity and potential, they reach the edge with more resources and they're better at it. They basically grow around everybody else. And the fun thing is to watch the fluxes. Watch the nuclei of the cells as a good tracker. On the outside is mechanically favorable to push your daughter cells outward. On the inside, near that perinecrotic boundary, you can either push against a dense tumor viable rim, or you can push a cell into that relatively hollow necrotic core. So you actually get a lot of cell flux pushing towards the center. And then in this necrotic stuff, you have cells shrinking but sticking together, and that actually matches what we see in organoid models experimentally. Is without something pushing this whole thing together, there's actually a lot of spongy stuff in the center in the necrotic core of a tumor organoid. So that's actually quite nice.
A philosophical question. So one has a model like this, it looks like the real thing, but is there any validation? Like you could say, well, let's assume we change this parameter. This is what's going to happen. We average out some behavior, and now it turns out in experiments, something like that.
So that's a great question. It gets down to really the core of modeling, right? Can we prove that it looks realistic enough to our training data and training assumptions? And then how do we validate that it can predict something that's outside its training set, basically?
Okay,
so one thing that we'll do is we'll say take the same cells, put them in different initial geometry, change the boundary conditions, or change a boundary condition somewhere in the middle of the simulation, or introduce something new, say inhibit something to mellow inhibitory experiment. And those kinds of things tend to work out. And I'll actually show an example where we got something like that.
okay.
This is a great question. Here we were able to calibrate to, breast cancer data in particular, get the right growth rates, and the curves make sense. And we also showed a lot of convergence work to say, as I change my delta T, do my results start to converge in measurables like, say, the tumor radius versus time, for example, and the cell density, viable ring thickness, whatever metrics we could come up with to show that this is like a numerically correct method, and that we could pick reasonable default values for delta T, for example, for the types of parameters we expect. Was there a question?
Is that, basically the interpretability of the model is important issue. So the result that you show that how is the behavior of cells, is it compatible with the biologist's interpretation of the mechanisms?
So if I'm getting that right, the question gets down to interpretability. One thing that is nice is that all the assumptions, and we're building towards this more and more, and I'll show towards the second half of the talk, is we're trying to use like human readable methods to write our model rules. And so it used to be that you would handcraft all the stuff like C++. It was very opaque, right? It gets to a point where even if you read the code, you may not know what it's doing. And so we're transforming. I'll show the second half of our talk. We actually have a modeling language we've developed that's human readable. And the model to us is the human language statements, which then get mapped into mathematics and turned into code on the fly as you compute. And so to us, the model is the structure, which is why this becomes a good knowledge model. Well, we assert it's a good knowledge model. We'll see what the peer reviewers say. And that helps at least to interpret, say, I know what's in this thing. Although they can turn out to be unstated assumptions that I think plague a lot of models. So Potts models and do self-sorting examples, for example. It turns out the random fluctuations of the cell plots are a vital part of the self-sorting. You don't realize that because it's cooked into cellular plots. If you do a center-based model, you can get self-sorting, but only if you add in some random vibration of the agents to model random subtle skeletal perturbations. And so that's like an unstated assumption in cellular Potts formulation. So we can run into things like that. Interpretability is a bit, so you have to say, what is driving the behavior of my emergent system? And we're just getting to the point now where I think it's easy enough to build models, that it's now reached a point where it's going to be easy enough to randomly delete rules. And see, so imagine like a deletion experiment, like biologically, you delete a gene and see if it made an impact. We're going to do the same thing with model rules. Say, delete a model rule, see if that rule was critical or not, and see if you can kind of come up. It's kind of like an information criterion, but more as a perturbation experiment in the model structure rather than perturbations in the parameters. I think we're just as a field reach a point where we can do that tractably, but it's wide open, I think. But that gets down to saying, what did it take to build my model, and what can I remove and still have the result that I care about? I think we're on the cusp of being able to do that as a field. Do you have a question? Okay, there's just okay, got it. So let me give an example. Oh, you have a question, good. Yeah.
What about the multi-scales?
I'm sorry?
How you deal with the multi-scales?
So I've hidden a lot of the details away, and I want to be careful on time. But we have actually a bunch of different delta T's. Because it's such a time multi-scale problem, I have the smallest delta T I have is for diffusion, because it's the most computationally difficult. After a ton of convergence testing and based upon our discretization, we found that we can get away with a delta T of 0.01 minutes for diffusion. Mechanics is the next time scale. 0.1 minutes seems to resolve most of the things that we care about. And then there's the phenotype time scale, like if cells are changing in the order of an hour, we use a delta T of 6 minutes. So I march along in time and say, not every process has to update at every time step. And so I have diffusion every time step, basically mechanics every 10 time steps, some of the cell processes every 60 time steps, and that accelerates our method a bit. And we did a lot of testing to make sure that that works as well, basically convergence testing. So that's one example of multi-scale. Another example is that those constitutive relations, right, the model rules, those basically taking smaller scale information like diffusing signals into the cell to make decisions about their individual behavior, and that scales up to become emergent systems behavior. And I think there's a lot of room to improve that too. We also have ways to incorporate, I don't have it in this talk, but we worked with with Institute Curie in particular. They had something called Modbus for Boolean networks. We combine Modbus for Physicell to create Physibus that can integrate a Boolean signaling model in each individual agent. So you can actually compute that for molecular processes if you care about it. We've integrated flux balance analysis models by basically approximating them with real networks and integrating those into the agents. You can do ODEs in there if you want to. And then the rules are like another way to add smaller scale information into the agents. So it becomes inherently multi-scale. So This kind of gets, the fun thing about biology is that everyone's right and wrong at the same time. So several years ago at a big cancer meeting, Physical Science Oncology Network in the US, we were studying hypoxia in particular, and we know that cells can adapt to hypoxia. They change their behavioral set. They can increase migration, decrease adhesion, change the cycling, change the metabolism. So you have this hypoxia adaptive phenotype. So there's a thought experiment. Take that adapted cell and take it out of bad conditions and put it back into good conditions, say normal hypoxia condition, normal oxygen conditions. So the thought experiment is, if you put that cell back, that adapted cell back into new conditions, how long will it take to revert back to its old behavior, if at all? And if you survey the entire room of biologists, what range of answers do you might expect from, what do you think is the right answer? Who thinks that it's instantaneous reversion back to old properties? Okay, we got a 0 on the time for how long it takes. Who thinks it takes on the order of seconds to revert to the old behavior? Okay, we've got a discrete spectrum. Do we have anyone who thinks it takes minutes? Okay, we've got minutes, 10 minutes, an hour, a day, a year, a lifetime. Okay, so okay, maybe not that much. But we basically got the whole right half of the real line when I surveyed a room full of biologists. This is a perfect place for modeling because we can ask this what if questions saying, can we model this? But it takes a combination of that plus something new experimentally. So Daniel Gilks at Johns Hopkins came up with this way to permanently mark hypoxic cells. They start off with fluorescent red. Then when they're in hypoxic conditions, HIF1 alpha builds up, and they snip out that red gene and now expose a green gene, and they permanently change color to green. So you can track to say who is either hypoxic now or has been. So you get the state tracking system. Now we have a way to visualize it, and it's time to build a model. So we basically built a very basic model that necrotic tumor that we saw before. I just modeled really that.
So experimentally, what is the answer?
That's an experimental system.
Yeah, but what was the answer in terms of how long it takes to readapt?
Oh, yeah. Well, we'll get there.
From the experiments, I mean.
In experiments, they were able to watch it in time tracking at that point in time. So they were able to image it like 1 day, 5 days, because it was a mouse model, and they didn't have the intravital imaging going at that point in time. So there was an open question.
They couldn't do it fine.
Not at that point, no. It was an opportunity for us. So,
I thought that was an in vitro.
yeah, no, that was in vitro, but we want to look in mice. So this is in petri dish.
Right, so in that.
That would have been a source of hypothesis. I agree. We might have been able to answer from in vitro, but we did want to care about in vivo. So that's a good question. Maybe we could have answered it without any modeling just by watching it in vitro to look at the switching times.
So it wasn't done with that data.
So this video,
Because there's gene expression time.
and I think
Maybe half hour.
probably could have guessed a priori a pretty good time scale. But what we wanted to do was to say when you're in actual in vitro and vivo conditions in a mouse or in a human, where you have more complicated gradients with not just a fast switch from one to another. Yeah, that's right. I think we probably could have gotten a lot more out of it than maybe we did at that point. But it was a good starting point for us. We built that necrotic tumor model I showed you earlier, and I added just a really basic model saying I either have a red gene or green gene, and red gene makes red protein, and I color it red. Green makes green protein, color green. So when we slip one, they basically kind of transition to orange and then to green. And we modeled that cells in hypoxia would have changes in migration and changes in proliferation. And then when they escape hypoxia, we did a sliding parameter said, what's my mean time to revert my behavior to basically go back to the red cell phenotype. So just basically one tunable parameter to investigate. And so if you model the instantaneous, you get this structure. Cell is here in the hypoxic conditions, I migrate, I reach happy conditions and I stop. Tumor goes a little bit, and my scientist moves, so I migrate a little bit more, and I stop. So you get the stable growing green pattern. And that would be great if cancer did that, because then we wouldn't have to worry about metastasis. So that's the pattern we got. That doesn't end so
Sorry, I don't understand.
the stable ring structure where nobody migrates to another organ to kill things sounds pretty nice.
But there may still be,
Oh, yeah, okay. It's been hyperbole. I agree. So, but a stable morphology would be a safer thing and easily surgically resectable compared to cells wandering all over the place. Okay, maybe it's a bit hyperbole. This would be lovely if it were true, but we would assert that it is not. Because we know that tumors don't just form stable rings. if you have a transient phenotypic, this is, that says, I migrate out, I leave my bad conditions and keep migrating a little bit further, and then I revert, and you get these invasive plumes. And interestingly enough, the stochasticity model says that there might be a little mechanical weak spot that one cell finds, they widen it, and everybody else follows. So it looks like collective migration, but it's just really every cell for itself plus stochasticity. And then if you have permanent change, so they never go back to that phenotype, they just keep wandering forever. And so we thought this is a little weird. We're actually very disappointed. We started looking for bugs in our code because we expected a little bit more uniform. And our biologist said, no, we see those plumes in our mice. And so out of all the hypotheses, that particular one created a prediction that is observed experimentally. So her imaging system was able to see this in mice for the first time. And we were able to give an underlying explanation to say these are the rules that create this kind of structure. So question.
Sorry, I might be a little bit confused. So the color, like, what's the link between the phenotype and the color of
So the red cells are the cells that have never been hypoxic. The green cells are either hypoxic now or have been hypoxic.
So you never switch back the color, you just.
No,
switch the
because that one
color.
permanently cuts out the red gene in her system. So we want them matched up in our model, so we can compare our observations to her observations.
So like, when you when you see the color,
When you see this green,
yeah,
This
those
is-
cells that have been exposed.
yeah, they either have been exposed, they have been exposed, either are hypoxic like these guys over here, or recently were. And so we get these growing plumes that are very similar to the structures that we compared, and we didn't see these in advance. So this is like an extra observation outside of our training set that we did not use to build our models. And so we were able to predict an emergent feature that wasn't in our observation set. So this is basically what we would say is moving in that validation direction. And since then, they went back and looked at the gene expression and found more evidence of it. So that's a published paper a few years ago, and you can actually try an online version of this model, kind of an early version of online pod modeling. Yeah, please.
So in your model, what was the time you used for switching back to aerobic respiration?
You know, we used a variety of them, and we found that this is fairly stable, giving the persistence time on the order of, more than just a few minutes. We gave a time scale on the order of hours to maybe a couple of days. But anything that was substantially above zero would give this plume. The shorter the persistence time, the shorter the plumes. The longer the persistence time, the more the breakout, which makes a lot of sense, posteriori. But we found that this kind of regime of non-zero, but finite, gave us the most reasonable behaviors that we saw, which might make sense in thinking about the time it takes to turn a gene off, decay protein products, get rid of mRNA, make new mRNA. That changes, new behaviors down the road. There's not an instantaneous switch in behavior. So it kind of makes sense. But we didn't have, we probably didn't have the smarts to think of it a priori. So we just investigate. So this is a relatively simple model that we're able to build. Proliferation, by the way, we calibrated KI67, motility to approximate Bayesian computing from migration tracks. We found that actually the red and green did not have hugely different changes in cycling, which surprised the hell out of us. But the biggest change was in migration. So they go and grow. So, thinking about what we can learn from this, I cut out an example because I wanted to free time. I think that was a good thing now, looking at this. We did a COVID model with Thomas and a bunch of other people, and every iteration of the modeling got slower and slower and slower because we started adding more complexity, immune cell rules, and it was all handwritten code. So that hypoxia model was pretty easy to code, although easy means it took less than a year. But every iteration, even with a team of 20 or 30, 40 people, slowed down to point. And I find that as the model complexity grows, it's like in 1 over n, the number of people can understand the code set until 1 over n is less than 1. And basically no one understands their project anymore and the whole thing dies. It's kind of what we found. So we need to get the preprint to paper.
Sorry, what's the bottleneck
I'm sorry?
The bottleneck in writing these big models? Is it in the actual coding or is it in the biology and experimental biology?
There are lots of bottlenecks. Actually, that's a great question. I swear I didn't plan for this question because it's perfect for like one of my next slides. But when you think about the key modeling steps, right, you have to formulate hypotheses, You know, how do biophysical signals drive some behaviors? You have to talk with biologists to figure out what those ought to be. It requires developing a common language to talk with biologists, right? We need to learn some biology, they need to learn a little bit of math, and you kind of meet in the middle. Usually mathematicians do a little bit more of the walking, in my experience. You have to turn the hypotheses into mathematics. You typipally do that manually for each hypothesis. And then, each of those hypotheses usually has to be written as code, typically artisanal handwritten C++ or Java or something else. And the more you write, the more you debug. So here's an example from a COVID model of all the hypotheses. We tried really hard to write them down cleanly because we're trying to be reproducible. These are the hypotheses that our consensus group came up with for macrophages in COVID-19. Each one of those 12 hypotheses was a bunch of code to implement. As you add more hypotheses, your likelihood of breaking one of the earlier ones with a code bug or some unexpected incompatibility increases. This is just one cell type in this COVID model with CD4s, CD8s, macrophages, neutrophils, oh yeah, M0, M1, M2, various weird other macrophage flavors. We had, God, what else? T-Rex. There's a zoo. I mean, that's the thing about immunology, it literally is a zoo. And this thing became difficult. We have the paper drafted, and then Michael Getz ended his postdoc, so sitting on my desk waiting for the final revision. But it really made us think about the problems of handwritten models, right? It doesn't do a good job of leveraging prior work because you have to learn manually from someone else's work, interpret their parameters, interpret their form, interpret what alpha 37 means in that parameter in X1 and things. We have a lot of plots and models in the math world that are not labeled, besides variable X, Y, Z, T2, 12, and 78, and V and U. So if you didn't catch the definition slide 4 slides ago, you have no idea what you're looking at, right? So this is a problem in biology and in math biology. Imagine that over many cell types. You have no idea what's going on, even with a legend sitting right in front of you. The harder it gets, the harder it is for people to get onboarded and participating and contribute to that project. And the likelihood of making error increases. And in particular, it gets more and more abstracted away from the people who know the domain. So it's hard for domain experts to contribute. And harder for us to do a reality check with domain experts say, is this complete nonsense? The thing you find in spaghetti diagrams that used to be popular about 15 years ago is where I put a rule in this diagram can break this diagram. And so it's really hard to decide where to place a new rule as this thing gets bigger. So let's keep that in mind for a minute. And then we did basic code profiling. Let me get a lesson from that. Code profiling, you said, I have this big computational code. Let's make it the same take so darn long. So you basically take a stopwatch and time the different functions and add them up and see where your bottlenecks are. So in our codes, we did this with Sunita Chandrasekhar in Delaware, and we found that we were spending like 65% of our time on the diffusion solver, which makes sense. Diffusion is hard, no matter how much you optimize it. And so we looked at just a decreasing marginal utility. If you can speed up your diffusion code by a factor of 10, 65% becomes 6.5% of your old time, and your new simulation time is like 40% of your old time. So, oh, that's great. I'm going to sink a bunch more effort in, and now I'm going to improve my compute time by a factor of 100 in my diffusion solver. Well, now we've gone from 42% to 36%. This is a classical economics rapidly decreasing marginal utility, right? For every extra dollar spent on code optimization, I get less and less out. Now, if I make my diffusion code 1,000 times faster, it probably took a factor 10 longer to get those improvements. And it only went from 36% to 35%. So, like, don't do that, right? Well, let's generalize this, right? Look for the biggest bottleneck, increase it by 1 or 2 orders of magnitude, and then move on. That's the lesson in scientific workflows. Code filing for a scientific project. How long does it take you to learn the biology and the background and the coding and the mathematics? Months or years? How long does it take you to develop a common frame to talk with biologists? If you're lucky, this is months. Right? You have to go back and forth and talk and say, okay, yeah, I got everything. Now you have to code it and debug it. This can be weeks or months or years, right? And then, calibration experiments is certainly a big bottleneck too, but this is a great problem to reach. Usually we're stuck somewhere around here. And then the simulations, everyone complains about how slow a simulation is. But what's minutes or hours here compared to all this stuff, right? So
Where's the going back to working with the biologists when you.
You're right, there should be a loop here, which may, I imagine there are loops within here, but you're right, this is just imagining one, this is just one pass when you think about it, right? So you should probably go back from here to here again, absolutely, and then hopefully you're learning all the time.
And typically, how many loops do you have? to
It takes a lot of loops, so this loop takes half a year,
No, I say, how many times do you have to go back to the biologist to re-examine your
certainly more than one,
experience?
right? I mean, multiple times.
Like the example you give.
Right, and it takes forever, especially because this time lag is so long, you get from here to here and say, I'm sorry, I don't remember this conversation over here. So you spend some time going back up to speed to say, let's recap, right? So it's really inefficient. As mapping and numerical analysts, we love spending our time optimizing here. This is like improving your diffusion solver by a factor of 1,000. This is where we need to spend our time over here. Right, and as mathematicians, it's very hard for us to want to do that because, you know, it's a linguistics problem, it's a language problem, it's communication. This is basically education too, right? You know, and all of the tenure reviews say do not care about that until you're done. Bad, right? So we want to create a computable model grammar that can truncate some of those steps a little bit, really smooth the way. And so our goal is that it needs to be human readable. I want to be able to make sense of it. I want my model statement in human language terms to be my model, to be equivalent, and that you paste that into the methods section, and people can understand what's going on. There are beautiful immunology models there that I, even as an expert in coding and okay at math biology, cannot understand and do not know what their assumptions are. I cannot reproduce results. Even if I read the chain of 5 or 10 papers that keep referencing each other, I can't find a clear statement of their hypotheses. So that's annoying, and it's not good science. So we want this, if we can succeed there, even just the process of writing down our assumptions, even if it's not computable, is really valuable because we're systemizing our knowledge in a way that we can transmit it along with our models, the assumptions. It's like the lost metadata of our models. Now, if we can readily translate these word sentences into mathematical forms, that's great. It's a little bit disappointing as mathematicians, but you notice, if you really look, you see a lot of repetition in models. There are only so many ways to write Fisher's equation, even with extra terms, right? I mean, you see a lot of repetition. Maybe we should just embrace that and move to the interesting bits instead of the repetition. And then we want to be able to integrate new rules with old rules so that new rules don't break old rules. So there needs to be some kind of a, you know, associative distributive kind of power in that. And then we want to be able to combine rules again in different ways, data-driven rules or knowledge-based rules. And so if we want to make a computable model of grammar that accomplishes this, sorry, I swear I'm getting close though. So we only have time for discussion. I'm glad I cut out about a third of my talk, by the way. So really, we need a dictionary of signals, like stimulate, symbols that mean something to us and to the computer, basically, so we can map them. We need a dictionary of reference behaviors to say, when I say migration, I mean this bias random migration with the mathematical form that I've written down in a really lengthy appendix somewhere. If I say secretion, I mean a specific source term in the PDE. When I say proliferation, I mean, you know, a stochastic transition and some certain things. So I need basically a library of reference behaviors, reference models for the sub-behaviors. Places where we're hoping is a little bit less controversial, right? Where we might agree on what those things mean, and then write them down so we don't forget what we mean. Then we need a grammar that connects these signals. Some signal changes some behavior with parameters. And then we need to be able to map that grammatical statement onto mathematics and then mathematics to code. So that's what we need. And so we started digging through, and in particular, the COVID model really drove us to say, we've seen a lot of models in the past 15, 20 years. Maybe we can find some common signals that a lot of people need. Diffusible factors externally, diffusible factors internalized. That's certainly important. The magnitude of the gradient is a good one. Concentration, say contact with a specific cell type. Say, I'm in contact with 4 CD8s. Okay, that's a signal. I'm in contact with 1 fibroblast. That's a signal. I'm in contact with 1 apoptotic cell. That's a signal. I'm apoptotic. That's a signal, right? Pressure. I have been squeezed in this way and I have a mechanical pressure, even if it's, say, dimensionless right now. Whether I'm alive or dead. And then any custom signals, say my language isn't complete, so someone might need to define a custom signal. Okay. And then I might look to say, oh, I better add that to the library, to the language. And then we have a dictionary of behaviors. Basically, all those processes models on the very beginning screen, everything we've learned, we started building in a standardized process model. So we have early ones that we've had for a long time, like cycling death transport, migration, chemotaxis, and mechanics adhesion. And we have new ones like transformation from one type to another, fusion with other cell types based on I'm in contact with someone I want to fuse with. Phagocytosis says I'm in contact and I have a rate of initiating phagocytosis, and eventually I just decide to eat it, I acquire its volume. Okay, basic conservation. Effector attack is fun because they say I am in contact with somebody, or I have a rate of wanting to attack it. It has immunogenicity saying how able I am to be recognized to be attacked. The combination of those gives me the success of initiating attack. I have a duration of my attack, and while I'm attacking, I stick to it by form of synapse, basically, and I cause damage to my victim. I'm sorry, to my target. Then, interestingly enough, whatever was written is actually two hypotheses. You have hypotheses on starting and doing the attack, but you also have hypotheses on what the damage does to you. So you need a separate hypothesis in the target cell that says damage increases my death rate. So you need to make sure to add both. And then we've added other things like a generalized damage. So you could say doxorubicin increases damage, so I can accumulate damage. We might say damage reduces transition, say, from S to G2 or something, or G2 to M to add a checkpoint. And then you might have a repair rate that says I repair my damage because I have Dance Revolution, DDR enabled in my cell, and then I can reduce my damage and continue with my cell cycle, for example, in a resistant cell line. So you can add all these kinds of things. Then you turn it into a grammar. Say, for some cell type, some signal increases or decreases the behavior. This feels stupid when you see it, right? It's ridiculously simple, but we wanted to keep it simple to see what we could get away with. I know we're missing things. I just want to get a knowledge model that can get 90% of the way there as a starting point and then refine. So, for example, for M1 macrophages, there's a T. The product cell debris as a signal, concentration, increases my transformation rate to an M1 macrophage. There's a model rule. Human interpretable means something biologically and hopefully does something in my code. Well, from malignant epithelial cells, doxorubicin increases death. So there's a classical hypothesis. And then in mathematical modeling, we basically need a response function to say that I go from a default parameter value in the absence of the signal, and then as I increase my signal, I should increase my parameter response. Well, there is a great function form that the PKPD community has used for 13 zillion years called Hill functions. We use those. I was so disappointed when I learned that there's actually named after a guy named Hill, and not because it's shaped like a hill. Learning moments. And so you can say, I should increase the cycle entry with parameters and write this as a response function going from your base value to your maximum response value. And then this is key. We made a multivariate version of Hill where we can add up all the up responses, add up all the down responses in a way that generalizes. If you go back to one signal, you recover your original Hill function. We add basically like a big OR operator. So there are things we need to fix, but this works. You can combine signals, and notice that the order doesn't matter. I can add a new up row and it doesn't affect it. I can add a new down row and it doesn't affect that. And then do a bilinear interpolation from the lowest thing you can have to the base to the highest thing you can have. So here's an example where I have oxygen increasing cycle entry and pressure, for example, decreasing cycle entry, and that combines into this kind of nice surface. And if you have a fixed language format, that means you can do a data format, which means you start writing support tools. So we wrote something called PhysicCell Studio, both a desktop version and a cloud version. In fact, here's an example of it. We're going to configure my environment, my basic parameters, diffusible things, cell types with all the base parameters, rules written in a drop-down way. Say, choose a cell type, choose a signal, what does it do, what does it act on, define Hill parameters, write the model rule. I can even visualize them. So I can plot my visualization in real time and then write it to a file and it'll parse all those things at runtime and create those mathematical functions. Then I can set my initial conditions and draw things. And later on, we're integrating with spatial transcriptomics now to say if I have something parsed, I can actually load it in and do that. So that's kind of work in progress.
There's a factory of this rules that then people would just.
Now that's side. Yeah, we want to get there. We just got notice of award yesterday. We were hoping to build that. We would love help. We would love help. Then run it here and then plot your results. So here is that basically that hypoxia model, a simple version. that oxygen crossing in, some don't escape and become necrotic, others, they've been exposed to low oxygen, transition to the motel type, and then skedaddle, and then they get far enough out that they transition back and start forming little colonies, for example. So there's a really basic, they basically played with this morning to build this model. So it's really easy to build. So with that, in fact, I did a 5-minute challenge at SMB to say, can I build a basic model in 5 minutes? And I actually put a clock on. So it's really fun and stressy. And I was sick too. So I made it with like 11 seconds to spare. So we can do this. And actually now, because you can do this in the cloud, you mean that people don't have to download anything. They don't have to install anything, and you don't have to write any C++, which means you can teach a multidisciplinary audience. So we've tested this at UCI. We've tested it at Northwestern. We'd love to test it with more people, but we're writing afternoon curricula now to say, let's introduce you to agent-based modeling as a concept. And actually, the next exercise we found that works great is I build a zombie model next, because it's fun and it exposes almost all the things we care about: transformations, attack, damage, secretion of chemokines, chemotaxis, cross out the words, and go for a break and say, now we're going to build a tumor immune model. And so you can get through a basic model in 1 day, which means if we run a workshop hackathon, day 1 is training, day 2, taper that training, and you have 3.5 days for your hackathon instead of 1 day for your hackathon. So we found that people can really do cool things. So here's an example we worked on just a few minutes here, where to a mathematician, this looks like a complicated model, a lot of assumptions. To a biologist, it looks stupidly simple, say there's no way this will do anything useful. And it's often the truth is somewhere in between. It's not useful and also too complicated. But, it's hopefully not that. So we worked with Lisa Cruz in particular at Oregon Health and Science University, Laura Heiser, and others. Joe Gray was giving rules. And we just wrote down simple tumor rules, M0 macrophages that start off naive, activate this, inflammatory macrophages, secrete chemokines that attract and activate naive T cells, become effector T cells. They start attacking tumor cells and causing damage. Damage makes them die. But then eventually the M1s become M2, secreting anti-inflammatory chemokines that start pushing these effector T cells into an exhausted state in a simplified model. And we just start, even without calibrating the parameters, just the model structure itself with order of magnitude parameters is reasonable. It recapitulates things we expect, which is a good starting point. Now we say, let's do perturbations to validate this thing. And that's what we're building now. We start off with a mix of naive T cells and naive macrophages. And I just randomly see the tumor cells. It forms a necrotic core, necrotic debris starting to draw on the macrophages. And you see some activation now to M1 macrophages, should be these yellow guys. And then they're starting to secrete and activate the T cells, so the magenta guys are attacking tumor cells. And then eventually we're starting to enrich for an M2 macrophages, and we're starting to really, so we kind of have an initial response and then it kind of dies down. So really simple model starts to recapitulate things we know, which says, rather than taking 2 years to build this model, I can build this model in an afternoon and get to the interesting bits like calibration and validation experiments. Some more advanced work with Alana's group is actually taking spatial transcriptomics and bulk transcriptomics and using those to identify the cell types, basically dimensionality reduction on the gene sets to identify who's there, and it's starting to do some of the spatial analysis to identify rules and to put those in. And so Alana's group did a great job of building some PDAC models in particular, calibrating some of the rules to what they were learning in their data sets. And I wish Alana were here to explain the results better, because they're deeper than I'm getting here. But they were predicting things like, if they can modulate some of the rules with virtual checkpoint inhibitors, they can start changing the interactions, and that would be matching additional observations that they got in experiments that weren't used to build the models. So you can do virtual therapeutic perturbations. The other cool thing was we were taking PDAC, cross-section them, segment them, identify them with, so right now the resolution of this is not single cell, it's clusters of cells, we kind of nucleate packets of cells, but it's getting there now. We're getting to a point of single cell resolution, especially omics. So we do this, and one of the neat ones was that actually some of the rules of fibroblasts, in context of fibroblasts with normal epithelial cells, can start kind of giving some transformation in their particular assumptions. And so depending upon the initial mix and configuration, you can get a tumor that was kind of encapsulated and stalled out, or you can get some tumors that were mixing with the right kinds and actually just exploded in growth. And so you can imagine trying to do that on an individual basis if you have more validating data, but that's like a proof of concept. So ultimately, we envision a lot of paths. Talk to experts, get some rules, talk to data, do some rules. We're doing some LLM, basically natural language processing, looking at PubMed to get rules, but they all get written in the same framework and they're mutually compatible. So we can start kind of building these things up. We have all this in a preprint, the modeling language, and actually it's in the PhysicCell release that's out there, and I'm putting together a new release that will be out pretty soon. The paper was reviewed once by Cell, and we have some revision plan with the editors, so we're hopeful they'll be out there. And as of yesterday morning, we got a notice of award to say we're going to take PhysicCell with the rules and start talking to the expert community to say, let's evaluate the rules, see if we need to extend the language, see if the language is expressive enough to do a few problems of pancreatic and breast cancer, and start coming up with consensus rules for macrophages and M1s and M2s and CD8s and CD4s and Tregs and all the terrifying zoo, but in a way that we can tractably go. And so the CoreGAPS is going to help us to get the positions, identities, and things like this for this basically a big dimensionality reduction technique. That's Alana's group. And we're building a cloud infrastructure to say, can I run the model in the cloud, save my results there, save my digital cell templates, and share them and query them with other people. So these consensus macrophages can come out. So then you can imagine, and this is where ITCO is funding this, at least I hope it's why they are, is a bioinformatician walks in the door, says, I have a spatial transcriptomics data set, feed it into the pre-analysis. It says, aha, you've got M0s, got M1s, you've got some pancreatic cells, you got this and this and this. Seeds them into the model, the right positions, and say, oh, now let's go to the library, find my M1 hypothesis, my 2 hypothesis, and it gives you a starting point. And we can go through. And we're connected to an HPC backend to start doing uncertainty quantification is the hope there too. So we'll see. That's what we're trying. So that's as of like yesterday morning. And so we'll be running virtual hackathons and workshops and things for this. And we would love help for coming up with the hypotheses. But we really think that this is kind of where things are going, to say this spatial data plus modeling to explain them. And writing down human interpretable rules so that the knowledge is really interpretable. Thank you for your patience and your time, and I hope that you enjoyed it, or at least don't throw too many tomatoes.